All right, welcome to part 2 of the cell organelles video. Let's dive right back in with lysosomes. So, lysosomes have these molecules called hydrolases. If you remember, when we were talking about macromolecules, we talked about the term hydrolysis. Hydrolysis, remember, is breaking macromolecules down into their monomers. Well, the enzymes that perform hydrolysis are called hydrolases, and you tend to have large amounts of them in these organelles called lysosomes. Remember, if you did not learn this in freshman bio, then you're learning it for the first time. The word lyse means to break or to burst. So the job of a lysosome is to break apart things, specifically to break apart larger molecules. It is the equivalent of a stomach for a cell because it can be used to break things down. In unicellular organisms, it can be used to digest food. In white blood cells, it can be used to digest bacteria or any other pathogen or disease-causing agent that's infected you. It can also be used to recycle any sort of damaged organelles. Let's say you have a mitochondrion that has been damaged in some way, it's messing up, and so you can basically shove it in a lysosome, have it digested, and reuse the molecules that are in there before, the mitochondria does damage to the rest of the cell or just, isn't functioning properly. So there are a couple of processes that are involved with lysosomes. Phagocytosis is basically you take in a solid particle of something, So if you don't remember endocytosis and exocytosis from freshman year, we'll go into those in more detail when we talk about cell transport, but basically part of the cell membrane encloses some form of food. It forms this kind of bubble surrounding it, and that fuses with the lysosome, and then it gets digested. There's also autophagy. Auto means self, phage means to eat. So that is literally a cell killing itself. It's programmed destruction, well, not itself. It's programmed destruction of certain parts of the cells. So if you need to destroy messed up components, you can do that here. You can also have lysosomes basically release their contents into the cell if you need programmed cell death, something we'll talk about later. So here we have kind of the process of phagocytosis being shown. You have something being taken in. Notice the membrane's forming this kind of bubble around it. That, when it's closed off, is forming this structure that's called a phagosome, literally means eating body, that then fuses or joins with the lysosome. The lysosome is going to digest it. You'll release whatever you need into the cell, and then the waste products will then be released from the cell. So if you're a single-celled creature, this could be how you eat. Similar processes happening inside other cells. So you see phagocytosis on the left where you're eating something, but then there's also autophagy where you are breaking down, unused or not unused, damaged or useless non-functional organelles. So one thing we saw on the previous slide was some mitochondria being broken down, so let's talk about them. Mitochondria are not the powerhouse of the cell. I do not ever want to see that written on any of your answers. The official job of mitochondria is they perform cellular respiration. So every time you say in class that mitochondria are the powerhouse of the cell, I'm going to stop you and make you correct yourself and say that mitochondria perform cellular respiration. And when you want to get technical about it, they are breaking down carbohydrates and transferring their stored chemical energy into ATP, which can then be used for cellular work. If you're looking at different cell types, if a cell has a lot of mitochondria, it is probably using a lot of energy. So one type of cell that has a lot of mitochondria tends to be muscle cells because they're going to need access to very quick energy breakdown, and they're going to need a lot of that access because they use a lot of energy. Mitochondria are one of a small group of organelles that have two membranes, very creatively named the outer membrane and the inner membrane. The inner membrane has a lot of folds. These are called cristae, and their job is to increase surface area, which we will talk about in more detail when we talk about cell size, cell membranes, and cell transport. There is a fluid-filled matrix inside, which contains not only ribosomes that are different and distinct from the ones in the rest of your cell, but it also contains its own DNA. So mitochondria are one of only 3 organelles inside of a cell that will contain DNA, one being quite obviously the nucleus, one being mitochondria, and the third being in plant cells, chloroplasts. Speaking of chloroplasts, they are actually a type of what are called plastids. These are found in plants mostly, some algae as well. Their job is to store things for the most part. What they have in common is they are all double membraned like mitochondria. They have their own independent DNA separate from the nucleus. They can have their own ribosomes, and they can actually, some of these can be kind of more general functioned, and some of them can differentiate into specific organelles with different functions. So chromoplasts are found in plants cells that have to have a specific color. So for example, here you see a flower. That flower has petals that are very bright to attract pollinators, and the way it gets that brightness is it has a lot of these chromoplasts, which are going to contain colored pigments in high concentrations. Leucoplasts tend to store starches, so you'll find them in, for example, the root cells of plants. They will basically be these kind of storehouses of this extra carbohydrate material. Chloroplasts perform photosynthesis. Honestly, you can just chloroplast function, photosynthesis. Mitochondria function, cell respiration. So chloroplasts also have 2 sets of membranes, like we said, and then they also have these internal membranes, or this internal membrane system called thylakoids. They're arranged in these stacks called grana. A single one is a granum. This is where light energy is converted to chemical energy, and then around the grana, in between that and kind of the inner membrane, you have the stroma, which is this aqueous. Aqueous, by the way, we'll encounter that term a lot, means watery. So this fluid around the grana, it's what has the ribosomes and the DNA. It's also where the energy, the chemical energy that was captured during photosynthesis, is used to make carbohydrates. And then these are all sitting in a matrix. A vacuole, its job is for storage. So most common place you'll find these are going to be in plants and fungi. They can be a storage place for waste products, certain ions. They are also potentially able to contain toxic compounds. So if you're a plant, you don't have the ability to run away from herbivores, and you don't want them to eat all of your leaves or all of your structures, because then as a plant you'll die. So one way that they can deter herbivores is by having either bitter tasting or toxic compounds stored in a vacuole where they can't harm the plant cell, but any herbivore that tries to eat them will either be poisoned or will encounter something really nasty tasting and learn to avoid that plant type in the future. Plant cells have a central vacuole where they store water. This water has a purpose. It kind of helps keep the cell what we call turgid, meaning full of fluid, and we're going to talk about the importance of that more when we talk about cell transport in Unit 3. They can also be useful to contain pigments and things like that in the reproductive organs of plants. In seeds, they can contain digestive enzymes, so that way it makes it easier for them to break down the stored fuel that they have. Freshwater protists, which are single-celled creatures, have these special structures called contractile vacuoles. They contract and they squirt out water, and this is a way to get rid of excess water. Once again, we'll talk about why when we talk about cell transport. And also you can say that food vacuoles are formed during phagocytosis, which, if you remember from lysosomes, is basically how unicellular organisms eat. So here we see a central vacuole in the center of a plant cell. It's usually kind of an open, empty looking space, but you can have things dissolved in the water that fills this central vacuole. And now let's move to stuff outside the cell. So there is an extracellular matrix that is usually released outside of the cell membrane. In eukaryotes, it's generally some sort of fiber and then a gel-like substance that these fibers are stuck in. So the most famous example of this would be the plant cell wall. I'd like to also point out that fungi have a cell wall as well that will have a different composition, but fungi don't tend to be heavily covered on the AP test, so we're not going to go over them here. The function of the cell wall is that it supports the cell, it supports the plant as well. Remember, plants don't have skeletons, they don't have bones, so part of the structure that helps keep them upright comes from the rigid material of their cell walls. Another important function of the cell wall is it limits the volume of a cell. And this is going to become really important when we talk once again about cell transport. They also form another physical barrier to help plants protect themselves against infectious bacteria or viruses. And they can help with form during growth and development. Once again, that just kind of talks about their role in the structure of plants. So they're semi-rigid, they're not completely unbendable. They're secreted or released to the outside of the cell membrane. A very large chunk of their volume is made up of cellulose. Cellulose is another polysaccharide, and it is made of a bunch of glucose molecules. Cellulose, if you're eating something like, say, celery or eating plant matter, that kind of chewy stuff that you just can never quite break down with your teeth, a lot of that is cellulose. It is not digestible by most organisms. In fact, the organism that's easiest able to digest cellulose is usually bacteria. So if you see anything that eats a lot of plant matter, chances are good they've got some symbiotic bacteria helping them digest the cellulose. The matrix is made up of proteins linked with polysaccharides, and you can have multi-layered cell walls as well. The extracellular matrix is found outside of plant cells. It holds cells together so that they form tissues and don't just fall apart. And they can also be very important to the physical properties of different types of tissue. For example, cartilage, skin, bone have different components in their extracellular matrix that give them their structure. Bone, for example, the extracellular matrix, a lot of it contains a hardened mineral made of calcium, which helps bones stay as rigid as they do. Cartilage, on the other hand, does not have that mineralized structure. It's got more stretchy, bendy protein fibers, so it is flexible, though it does also provide some support. The extracellular matrix can also help form a filter. So in the kidneys, for example, the extracellular matrix can be helpful there. And it's also kind of not quite like a signal, but it helps orient tissues, orient cells when they're moving around. There's a lot of cell movement that happens during development, during fetal development, and the extracellular matrix can kind of help guide those cells so they get to the correct location. You can also do this during tissue repair as well. So the fibrous component is mostly a protein called collagen. It's got then proteoglycans forming the matrix. The proteo, of course, means that it is made of some amino acid elements, and the glyco, of course, means that it's got carbohydrates in it. So these are more of those kind of what I call Frankenstein molecules. And then there are other proteins that link that collagen and the matrix together. Collagen is kind of flexible, but it's not really stretchy, so it's a good material to hold stuff together. Here's just another image. You also are seeing a close-up of a proteoglycan down here. A proteoglycan has proteins and associated carbohydrates attached to the outside. So here you see the extracellular matrix sticking to the outside of a cell. So this is the cytoplasm, this is the extracellular part, and you'll notice it connects to elements of the cell membrane, which are then connected to elements of the cytoskeleton. So that way you can have cells attached to this extracellular matrix without, like, ripping parts of the cell membrane off. They can also be used to connect, to basically connect and hold cells in place. This is done by a class of molecules of proteins called integrin. Integrin basically can change shape, so it can hook onto the extracellular matrix. And if you have cells that are supposed to move, for example, white blood cells will do this when they're rolling around inside your blood vessels, moving from place to place. The integrin can then change shape and detach, and then allow the cell to become free. Notice the integrin is not only stuck through the cell membrane, it's also anchored to the cytoskeleton. Once again, so you're not kind of ripping out chunks of cell membrane, you're firmly anchored. Cells can also have these openings connecting them. Well, not necessarily openings, these structures connecting them called intercellular junctions. This can help them stick to each other, or it can, in some case, even form tunnels in between cells. So, common one in plant cells are plasmodesmata. Plasmodesmata are these little channels that basically make the cytoplasm of cells that are adjacent or right next to each other continuous. So let's say that you start out your some molecules over here in this cell, you can actually travel through the plasmodesma and you can move to the next cell without having to pass through a pesky cell membrane or a cell wall. So there is continuous, like, cytoplasm through them. This can allow water, ions, hormones, even some small RNAs, some proteins to move from cell to cell. Animal cell junctions are kind of different. They're glycoproteins, which, once again, sugary glycoproteins. So they're more of those Frankenstein molecules. They will help cells stick together, and they have different functions. Tight junctions are, I kind of think of them like rows of staples holding things together, although you might think of them like really closely sewing things together. They form these boundaries, they form these connections between cells that will not let stuff pass through. So you're going to have tight junctions between cells when you want to have some sort of waterproof barrier. So for example, lining your intestines, lining your skin. Desmosomes will link cells to each other, but they will still allow stuff to move between those cells. So any cell layers that you want stuff to be able to flow through or flow between, you're going to have held in place by desmosomes. And gap junctions are kind of like plasmodesmata in that they allow stuff to move directly from one cell to another without having to pass through the cell membrane. So you can have actual molecules that are transferred from one cell to another directly. They're not super common in adult cells, though they're found a little more often in embryonic cells. And this is just close-ups and some electron microscope images of these types of cell junctions. Other electron microscope images, feel free to pause and check them out. So that enormous body of information was the information on cell organelles. Please watch this video multiple times, because if you don't understand what organelles do, or you don't understand some basics about their structure, it's going to make what we talk about not only in class but in other lessons very difficult to understand. Thank you and good